In this talk, we're going to learn about the different steps involved during viral infection. Viral infection involves many different steps, including attachment, penetration, uncoating, biosynthesis, maturation and assembly, followed by release. Viruses are considered as obligate intracellular parasites because they need a host in order to replicate. They replicate inside the host and hence they are intracellular, and they are parasites because they provide no benefit to the host they have infected. Whether a virus is living or non-living is controversial, but for the most part they are considered as non-living particles because they cannot replicate on their own. However, it should be noted that in spite of being considered as non-living, they can cause a lot of sickness and misery. Viruses are considered inert until they come in contact with the appropriate host. One thing to remember is that the virion is a term that is used to define a single mature infective viral particle. When we look at the composition of a virus, they normally comprise of a protein coat called as a capsid, and this capsid covers the nucleic acid that is inside the viral particle. There are many steps involved in the viral infection process. The first step involves the attachment of the virus to the host cell. This is usually mediated by host proteins present on the host cell surface. Once the attachment has occurred, the virus has to penetrate inside the host cell. Once it's inside the host cell, the protein coat has to be removed so that the nucleic acid can gain entry into the host. Once the nucleic acid has access to the host, biosynthesis has to happen where different viral components are synthesized by the host cell. This includes new viral nucleic acids as well as different viral proteins. After the biosynthesis step, there has to be a maturation and assembly of the new virions, and finally, the release of the new virions into the environment. This release can be in the form of a non-lytic release, where the virus buds out and the host cell survives, or it can be in the form of a lytic release, where the host cell undergoes cell lysis. The first step in the infection process is the attachment of the virus to the host cell. Viruses encounter host cells through random collision events, and thus they do not actively seek out the host cell. Let us take an example of intestinal cells that are shown in this figure and look at how they may get infected. These epithelial cells can have two parts. The upper part is called as the apical surface, whereas the lower part is called as the basolateral surface. Viruses usually encounter epithelial cells, and the viruses that attach to the apical surface of these epithelial cells are known to cause localized infections. However, if a virus is able to get transported to the basolateral surface of the epithelial cell, then the virus can then spread to other sites, and thus it can be a more widespread infection. The host membrane must be able to have proteins that can attach to the virus, and thus host receptors enable attachment of the virus. As shown in this figure, we can see the host receptors that interact with viral components and hence facilitate the intake of the virus. An example of such a virus and host protein interaction is where the SARS-CoV-2 virus's spike protein can bind to a host protein called as the angiotensin converting enzyme 2 or ACE2 protein. This interaction facilitates the entry of the virus inside the host cell, and this entry is normally receptor mediated. The second step in the viral infection process is penetration of the virus into the host cell. This is followed by uncoating of the protein coat of the virus, which allows the viral genetic material to access the host cell. Penetration allows the virus to go inside the host cell, while uncoating allows the genetic material to access the host cell. Uncoating can happen in different ways depending on the virus. Here's an example where we can see the uncoating of the virus occurring during the attachment process at the plasma membrane. In such cases, the penetration of the virus as well as the uncoating step where the nucleic acids can now access the host cell have been coupled together in one step. In another example, the virus can be internalized inside the host cell in an endosome, and once this internalization or penetration has occurred, then the uncoating step can occur. Another example is where the virus is able to enter inside or penetrate inside the host cell, and from the endosome, it is able to access the cytoplasm of the host cell and dock onto the nuclear membrane, and that is where it gets uncoated or loses its protein coat so that its genetic material can directly enter into the nucleus. Penetration is an important step in enveloped viruses as well. These viruses have an outermost envelope that they derived from the host cell that they had infected previously. This envelope helps in fusing with the host cell membrane much more easier. Now, depending on the virus, uncoating can occur at different stages of penetration. We can have an example where the virus penetrates into the host cell, and in the endosome, the viral protein coat and envelope is able to fuse with the host membrane and release the viral nucleic acids into the cytoplasm, thereby uncoating occurs. In another example, instead of the entire viral membranes fusing with the host membrane, a pore can be formed which allows the uncoating and the transport of the viral nucleic acid into the host cytoplasm. In another example, we can have the enveloped virus interact with proteins on the plasma membrane of the host cell, and fusion can happen directly in the plasma membrane, thereby allowing the nucleic acids to go inside the host cell. During the process of viral infection, it is important for different viral components to be in the appropriate parts of the host cell. For example, viral nucleic acids must be transported to the nucleus of the host cell for a successful viral infection. This can be done in a variety of ways, and one way is where the viral nucleic acids use host proteins that mediate the nuclear import of the virus across the nuclear pore complexes that are present on the nuclear membrane of the host cell. Another mechanism that can be used is where the host proteins are able to transport the intact virus across parts of the nuclear pore complex, and after they encounter a certain part of the nuclear pore complex, uncoating can happen, thereby allowing the viral nucleic acid to gain access to the nucleus of the host cell. Another mechanism employed is where the viral particle is small enough where it can directly pass through the nuclear pore complexes and thereby enter the nucleus. Once in the nucleus, the virus can undergo the uncoating process and thereby have the viral nucleic acid directly present in the nucleus of the host cell. In another mechanism, the viral particle can dock onto the nuclear pore complex on the cytoplasmic end, and on that side, a pore can be formed where the nucleic acids can be threaded through the nuclear membrane to gain access to the host nucleus. In the case of some viruses, the nucleic acids are present in the cytoplasm of the host cell, and they gain access to the nucleus of the host cell only when the host cell undergoes mitosis. During the process of mitosis, the nuclear envelope will dissolve, thereby allowing the viral nucleic acids to gain access to the host DNA, and once the cell undergoes mitosis and new nuclear envelopes are formed, the viral DNA is now, or RNA, is now present inside the nucleus of the host cell. Once the virus is encoded, it is able to hijack the host machinery to make new virions. For this to happen, different molecules have to be synthesized, and thus biosynthesis is an important step during the viral infection. The different molecules that have to be synthesized include nucleic acids, which could be DNA or RNA depending on the type of virus, or proteins. These nucleic acids and proteins play important roles in the formation of new virions. One thing to keep in mind is that during the biosynthesis of viral components, the host transcription is inhibited, and thus the host genes tend to be suppressed from being expressed or transcribed or translated. The newly synthesized viral components that are synthesized by the host cell need to be transported to specific sites in the host cell to promote virion assembly, and this step is called maturation. Microtubules, which are cytoskeletal fibers, are known to play important roles, as shown in this figure, in the transportation of different viral components to the site of synthesis. Once maturation occurs, virion assembly can occur. The different steps involved in the assembly of virions includes the formation of different structural subunits that form the capsid and the actual assembly of the capsid. Once the assembly of the capsid is underway, the viral genome has to be packaged within the capsid. The assembly of the viral envelope, glycoproteins, which are proteins that are present on the viral envelope, needs to be assembled in the case of enveloped virus. Thus, many components have to come together to form the new virions. During the assembly process, the viral genome is not transcribed or is replicated, and this ensures the efficient assembly of new virions. The location in the host cell where the assembly of the virion occurs depends on the type of virus. Enveloped viruses tend to be assembled at sites near membranes, like the plasma membrane, or near an organelle, and this depends on where the envelope of the virus is derived from. On the other hand, non-enveloped viruses can be assembled in the cytoplasm or in the nucleus because they do not need to be near a membrane to obtain the envelope. Once virions are assembled, they have to be released from the host cell. Thus, the final step of viral infection is the release of these newly formed virions. The release of virions can be through the lysis of the host cell, and in this case, the host cell membranes burst open to release the viruses, and hence the host cell dies. On the other hand, another mechanism of release is where the newly formed virions can bud out from the host cell. This is normally observed in viruses that have an envelope and involves different steps. The steps involved in the budding process include the formation of a bud, as shown in the figure. The bud that is formed then starts growing, and hence bud growth occurs. As the bud grows, it becomes large enough where fusion of the bud membranes has to occur. Once this fusion occurs, the newly formed virion is now able to separate from the host and thus is released. With this, we come to the end of our talk, where we learned about the different steps involved in viral infections. that include attachment of the virus to the host cell, penetration of the virus into the host cell, uncoating of the protein capsid to allow nucleic acids to have access to the host cell, biosynthesis of the viral genome, as well as different proteins that are needed to assemble new virions, the maturation and assembly of the new virions, and finally, the release of the virions from the host cell.